blue sky though. Oh, that breeze picks up. All right, so I don't have the stuff back from the printers yet, so I photocopied the first few sheets. We'll just probably have time to talk about true anemias rather than all the white blood cells as well. So we'll pick that up on Tuesday, along with blood tests and blood typing and all of that. So if you have any extra time over the weekend when you're not sleeping in and catching up on your sleep, usually blood typing is what gives students the most trouble in the unit on blood. We will have a quiz at the end of lab on Tuesday on identifying white blood cells. So you'll have time on Tuesday during lab to practice that. But those are the two things if you want to have any extra time over the weekend and you feel lost without something to do. Work on that. All righty, so remember we talked about blood and we talked about the plasma and the blood cells. The way in which this is determined is not usually done with that large a tube of blood, but with a smaller tube that looks like this, called a capillary tube. And I have some here in a jar. We quit doing body fluid testing about 15 years ago. So it's really unfortunate because students really enjoy wet labs where you guys get to do things with your hands and finding out your own blood values, your own urine values even. What's your hematocrit, what's your clotting time, what's your blood type. When we put to just illustrate why they're called capillary tubes, is that blood is taken up into the tube via capillary action. So you get your finger poked. Nowadays they would draw blood from your elbow and then just take it out of the tube, but in lab we would do finger pokes. And get a drop of blood on the surface and then just place the open end of the capillary tube in there and just draw it up by capillary action. So you're not sitting there sucking blood up from the tip of your finger. Okay, so I thought I'd just pass these around for those of you that haven't seen them before. They're real fun to play with in a Bunsen burner, like a glass pipette. You can heat them up and then you can stretch them out until they're almost the thinness of a hair. But so you draw the blood up and you want to get it past 3/4 of the way. And then, of course, if you put it in a centrifuge, you don't block it off. It's just going to completely flow out of get blown out of the capillary tube. So tablets of clay, just like modeling clay or something, are provided, and you take it, you push it down into the clay, let it twist it a little bit, and it puts a little core of clay at the bottom of it, and that plugs it. And then you would place it in a flat centrifuge. It doesn't need to be, it's not as large as the previous test tube. You just place it in a flat centrifuge like that, spin it for about 3 minutes, stop it, and you should have a lid on top that screws down over these in addition to that one. When it comes out, it will have pulled the heavier elements towards the bottom of the tube. So as it's spinning around, just like going around the corner of while you're driving, my kids are, in high school and college now, they still like it where we have 2 turns right before our house and they'll just all of a sudden yank the car over really hard, throw them against each other. The simple things that kids enjoy. So that pulls the heavier things down to the bottom. Those are our cells, but we actually talk about them as formed elements. All right? Anybody have an idea why we don't call the whole thing just cells? Why do we use the term formed elements? And the clue is in, well, I'll get to it in a second. This picture. This is a microscope side of blood in which the cells are whole cells, are not sections.
Some of them aren't complete cells.
Correct. Correct. They're not complete cells, mostly because of the platelets. Platelets are small sacs of membrane that pitch off of a very large cell, so large it can't leave the red bone marrow. But even the red blood cells are not complete cells. They don't have a nucleus anymore, much of their cell organelle. So they end up, by the time they're in the blood vessel, the vascular system, they end up just being membrane sacs of hemoglobin. All right, they're more of a cell than the platelets are, but that's why we use the term formed elements. So when we look at plasma after we spin it down, these are the proportions I gave you for the formed elements versus the plasma. And we get this, you'll do this with the lab exercise, by just doing a ratio. Instead of doing volume, it's a ratio of distance or length. So using a millimeter measurement, measuring the number of millimeters it takes for the blood divided by the cells, formed elements, by the total amount of length that the blood occupies. Not to the end of the capillary tube, but to where the end of the blood is. And that will give you proportions that can be assumed to be volume, and this is known as hematocrit. Right? So on the lab quiz on Tuesday, in addition to identifying the white blood cells, you'll need to know some of these blood tests. Okay? So the hematocrit is expressed in percent, 45%, 32%. Remember, it's lower in women. So, and you'll see different values, but in women it's approximately 38 to 42%, and in men it's approximately 46 to 52%. You'll see a range lower or higher, but give or take 2 or 3%. Okay? So the factors that change the hematocrit are the same factors that change the viscosity of blood. All right? So as viscosity of blood goes up, hematocrit's going to go up because it's really a measurement of that. And as viscosity goes down, let me reverse it. So as viscosity is increased, hematocrit is increased because you have an increased amount of cells, and as viscosity decreases or the blood becomes more fluid, then hematocrit is also going to go down. Now, looking at the content of the blood, the plasma is that fluid portion that doesn't contain the formed elements, and here are the primary proteins that it contains. Obviously, it's going to have ions and minerals, but these are the primary proteins, and the reason I have two different colors on there is if we take plasma, so we spin it down, spin the blood down, and remove all of the blood cells from that, and then take away the clotting proteins, the resulting fluid will stay liquid. And that's called serum. All right? So in emergency rooms, they store anti-snakebite venom serum, which is blood from large animals, usually horses, that have been exposed to the toxin of various types of snakes, and the serum collected from that, it would contain antibodies against that toxin. And we'll talk about the reasons why we would use that next week. And this could be stored, all right, without any anti-clotting agent. If you just take this plasma and store it in a tube without the blood cells and without the platelets, it will still clot, all right? As we'll see next week, clotting doesn't require any cellular material. My favorite biological material is Jell-O. I use it to illustrate everything from connective tissue, cartilage, to clotting. So when we have Jell-O and we add hot water to it, it's very fluid. You pour it, feels just, acts just like water. But as it cools off, it becomes a solid or semi-solid, depending on how much water you add to it. And what's happened? What happened between the heat and the cooling off process? Now, Jell-O is probably 80% sugar, but the rest of the material is protein. And as it is heated, it changes its nature and it polymerizes into long, feathery type molecules that trap water between the protein filaments. And when water is trapped and doesn't flow, it has a characteristic of a solid. Right? So with clotting, the clotting proteins are dissolved in your blood, but when triggered, they polymerize and form long filaments that interweave like felt and trap water of the plasma, and we have a clot. Right? Platelets can trigger this to occur. Platelets can be trapped within the fibers, but they're not required for a clot. All right? Platelet plug and a clot are two different things. All right, so what are these proteins? Albumin is the most abundant one, all right? Made by the liver, and it functions to maintain osmotic pressure. So somebody who has liver disease or is starving, doesn't have enough protein in their diet, has a distended abdomen, it's not fat, it's extra fluid. You may see pictures of, kids in Haiti that are starving or from some other country, and they have really skinny arms and legs because they don't have enough protein to make muscle, but large distended abdomens because without the albumin, the fluid leaks out of the capillaries and fills that abdominal cavity. Globulins are transport proteins made by the liver. We'll also be talking about immunoglobulins, which are not made by the liver, okay, and are made by lymphocytes, one of the blood cells that we'll be talking about in the second half. So this unit combines blood cells and the immune system. And the immune system, the specific portion of the immune system, is based on lymphocytes, one of our 5 types of white blood cells. Then we have the clotting proteins made by the liver and other molecules, enzymes, and hormones and ions and other components in that fluid. All right, most of the formed elements are red blood cells. By number, red blood cells are in the millions, and our anatomical, scientific, medical term for red blood cell are erythrocytes. Erythro meaning red, okay? And by number, we have about approximately 4.5 to 6, or you might see 4 to 6.2. Again, there's a little bit of a range. Million red blood cells per cubic millimeter. Okay, that's not a that's not a milliliter. A milliliter would be a cubic centimeter. All right, so this is a cubic millimeter. One of those tiny little squares that we had on our ECG, the tiniest of the square, make that into a cube, that's a cubic millimeter, okay? So 4.5 to 6 million erythrocytes per cubic millimeter. White blood cells, which we'll talk about on Tuesday, range from 5,000 to 10,000. So they're quite a bit fewer. And platelets range from 450 to 600,000. Okay, so they're kind of in between, but they're really tiny. So the process by which blood cells are formed is known as, you can see the comparative number of white blood cells, and usually we only see 1 or 2. So this process of forming blood is hematopoiesis, and specifically red blood cells are erythropoiesis. So the sites that we find hematopoiesis occurring in the fetus are more numerous than in the adult. So in the fetus, new red blood cells or new blood cells period are made in the liver. That doesn't happen after birth. They're made in the spleen. Instead, after birth, the liver and the spleen are where we break down red blood cells. Okay? They're found in long bones, finger, your humerus, your radius ulna, femur, and they're found in flat bones, the bones of your cranial cavity, scapula, sternum, ilium. Okay. In the adult, and I'm talking about 4 or 5 years old and on, we find them in flat bones primarily. And the previous slide with a picture of the calvarium, the top of the skull. And so this is the inner surface. And we have a thin layer of compact bone, inside and outside, and then spongy bone. Anybody learn what that was called when you were in Bio 430? Pardon? Diploid, yes. So, okay, and that might be one of those short words at the end of an exam. Another name for the red marrow and the spongy bone of the skull is All right, so flat bones, primarily the site where they're actually obtained from, or bone marrow testing, or transfusion is the sternum and the iliac crest. We don't go boring holes into people's skulls to get their bone marrow. And the scapula is a little too thin for that as well. So when I was teaching at the med school, we would have residents that were working in the emergency room coming over to the lab and doing sternal tasks, practicing on the cadavers, getting bone marrow out of the sternum, because it's a much quicker and faster. You don't get as much as the iliac crest, but it's a quicker way of doing it. When we look at long bones, remember the two major regions of a long bone? The two ends are known as the epiphyses, around, and the long shaft is the diaphysis. And in the fetus, the diaphysis is filled with spongy bone as well. There's with red marrow. In the adult, only the epiphyses have the spongy bone, and the shaft is hollow and has the fat or yellow marrow. So in the adult, it's long bones, but only the epiphyses, okay, which is plural for epiphysis. All right, so that's where hematopoiesis occurs. And when we're looking just at red blood cell development, this is known as erythropoiesis. White blood cells would be leukopoiesis. And here's a chart that follows from a stem cell. So a stem cell is just going to become a white blood cell or a red blood cell or a platelet producing cell, maybe a karyocyte. We don't know which. All right. And then the nucleus starts becoming transcribed as messenger RNA for a specific protein is made. Well, the enzymes that help synthesize that as well. Anybody know what that protein would be? What's the primary protein of a red blood cell? It's too obvious. Hemoglobin. Okay? Globin is actually the protein, heme is the iron content of it. And so, see how much darker and purple this is? That's because basophilic stains are particularly attracted to ribosome material, RNA material, and so it looks really dark here. And so you don't have to follow the entire sequence, but I do want you to know that erythroblasts are the stage at which we can determine that it's going to be a red blood cell and not, the proerythroblast is the state which we can determine it's going to be a red blood cell and not a white blood cell. Okay? The erythroblast stage is when it actually starts the process, but proerythroblast, we know it's going to be a red blood cell, not a white blood cell. Now, you'll notice that as the messenger RNA is used up and the ribosomal RNA is broken down, the cell becomes more and more pale. So it's losing its ribosomal material and gaining in hemoglobin. Until finally, usually before it enters into the blood vessels, the nucleus is extruded, and we end up with a cell that is just a plasma membrane sac of hemoglobin. Now, sometimes the body is under stress. Increased need of oxygen, either because of moving to high altitude or disease or extra activity. And so the erythropoietic process is stimulated to occur so rapidly that the red blood cells are pushed into the blood vessels before they have time to completely mature. And strands of ribosomal ER are still present, and so that's called a reticulocyte. You see these little lines. But normally, it appears as a mature red blood cell. This is just a picture of bone marrow. We have all sorts of white blood cells and red blood cells. Most of these are developing red blood cells. So a red blood cell, we already talked about in a capillary size, is approximately 7 microns in diameter. And notice that it's not spherical in the sense of being ball-shaped. It is round, but it's not ball. It has what is identified as a biconcave shape. Now, what's the purpose of a red blood cell?
Carry oxygen.
Carry oxygen. It will also actually carry carbon dioxide, but its major purpose is to carry oxygen, okay? And so if we were to utilize the basketball concept, and it's in the lungs picking up oxygen, the hemoglobin that's in the center of the cell, that oxygen would have a long distance to travel into and out of when it's picking up or delivering the oxygen. So that's not very efficient, all right? And so the biconcave shape, which actually is a very unique feat of mechanical engineering, while the crosslinks on the deep surface of the membrane hold it in that shape. And so no matter where the hemoglobin iron components are within this cell, they're relatively close to the surface, okay? And there's a high surface area to volume ratio. So the protein is hemoglobin, two types, alpha and beta, and within each one is an iron atom. Now, when iron forms an ion, does it lose electrons or gain electrons? Does it have positive charges or negative charges?
Gains electrons.
So an iron ion is typically written like this. All right, it has a variable number. It could actually go up to 3. Positive charges, so it loses electrons. And oxygen is an electron hungry. Oxygen only needs one extra electron to complete its cloud, all right? And so it's going to take quickly the electron from the iron atom and form a weak ionic bond. Why is it important that this bond be weak?
So the oxygen.
Exactly. We would need the oxygen, if it binds tightly, then it's just going to go in circles around the body and never be given off to the tissues. All right, so when we get to the respiratory system, you'll see how the affinity changes. I'll just give you an introduction to the oxygen dissociation curve, which looks like this. This is the binding affinity relatively between oxygen and iron. So this is when we have 100% of the hemoglobin saturated with oxygen, and this is increasing amounts of oxygen in the blood. So in the lungs, there is up to about 85 to 100%, or 100 millimeters of PO2. We can saturate the lungs 100%. So that means that you don't have to stay at sea level to keep your hemoglobin saturated with oxygen. You can go up several thousand feet and still get 100% saturation or close to it. So it would be very unhealthy, however, if we had high affinity between hemoglobin and oxygen, and it stayed at that. So even when we drop the amount of oxygen to 0, dissolved in our fluid, it would never come off. That wouldn't do us any good, okay? And if we had our line down here, then no matter how much oxygen we had dissolved in the tissues, even if you were breathing 100% oxygen, we would only saturate the hemoglobin. We would be able to, up to maybe 20%, we wouldn't be able to transport very much of it. So with this curve, we have a plateau where we can saturate the hemoglobin in the lungs with high amounts of oxygen available. And when we get to the tissues, and we start having excessive levels of hydrogen ions and CO2 because of activity, those conditions change the interaction between oxygen and iron and hemoglobin, and it comes off much faster, which is what we need it to do. All right. So this next animation shows the change in the shape of hemoglobin when it's oxygenated and when it's not oxygenated. So this change is affected also, like I said, by pH and by CO2. It actually changes the shape of hemoglobin, so it makes it more or less likely to stay bound to the iron ions and come off or stay bound to them. Okay? Well, here's a little bit of a diagram of the life cycle of a red blood cell. So it's produced in the epiphyses or the diploid and enters via the sinusoids into the cardiovascular system and lives about 120 days. That's about 4 months. Does it reproduce in the blood vessels?
No.
In the blood? No, why not?
No.
No nucleus, okay? And does it make more hemoglobin once it's out there? Still no nucleus, so it can't make any more messenger RNA. So it's basically, like I said, a bag of hemoglobin. It's broken down in either the liver or the spleen, and macrophages in those two organs will release amino acids and iron that can be recycled to some degree, and a pigment known as bilirubin. That pigment is excreted in the body via the small intestines and into the bile, right? So it adds a yellow brownish color to fecal matter, or if someone is throwing up so much that they start throwing up intestinal contents, then it adds that yellowish color to emesis, or it's also processed and excreted by the kidneys, and adds a yellowish color to that along with the urea. So, bilirubin in excess provides a yellow color to the skin and the sclera of the eye, and this is known as jaundice. So jaundice occurs, you might have a normally functioning kidney, but too many red blood cells being broken down. So if someone has ABO incompatibility, maybe the mother is Rh negative and the baby is Rh positive, all right? Or the mother is O and the baby is A, then antibodies from the mother can cross into the placenta and start destroying the baby's red blood cells. That can be so severe that the baby dies before it's born, or the baby can be born and be very jaundiced, okay? Newborn babies often become jaundiced. In fact, when babies stayed in the nursery for several days, they would become jaundiced while they were still in the nursery. So, is it still 24 hours? It was 24 hours when my kids were born. When I was born, well, my sister was born a year before I was, my mother had to sign her out at 10 days. She was like, I can't stand this food anymore, I'm going home, and she had to sign a medical release that she would take full responsibility for taking her infant home at 10 days. 10 days. Well, we were living overseas, and she wasn't really, it was an English clinic, but she told my dad, she said, they're bringing me the same food every meal. And my dad was, oh no, they wouldn't do that. So we put a little mark on the plate, sure enough, the next meal there it was. And she was like, I've had it. So, even within 3 days, you can start to see some jaundice. All right. Now, my twins, my son came home after a week. My daughter came home in 24 hours. And so I would take them outside when I got home from teaching. I was teaching 5 units at 4 different schools. And I would just, my mom was there taking care of them. They weren't home by themselves. And I would get home about 10:30 from Sac State on Tuesdays and Mondays and Wednesdays and Fridays. And I would take one kid at a time. They were born in September, and I'd just go sit in the lawn chair, take their clothes off, and stay out there for about 10 minutes, and then go back in and take the other one in. Ultraviolet light helps in the skin to break down excess bilirubin that's in the blood vessels. It's strong enough to penetrate through the skin. And this was found to be the case almost serendipitously by nurses who noticed that infants along the window were less jaundiced than infants that were deeper into the room. And so now we use Billy lights. And the babies will be in an incubator with a kind of their own little tanning chamber. And they'll have little black eye covers. And you'll see them little propped on their stomach with the little bottoms up in the air and kind of a diaper lightly covering there so that they can be exposed to the energy of the ultraviolet light. Alrighty? So liver problems, liver disease, or excess numbers of red blood cells broken down by antibody interactions either in the adult or in the infant will cause that to occur. All right, let's just look at a few examples of anemia. All right, anemia means too few red blood cells. What do you think is the primary symptom? Sign, I get them all confused. primary indicator, just avoid the words altogether. There's a particular rule for it. It's like, one is fever and the other is one is a measurement and the other is how you feel. Like nausea is a symptom, a fever is a sign. So this is something, a symptom, all right? So what's a symptom? What is a change in how you feel? Fatigue, exactly, because the function of red blood cells is to transport oxygen. So would bruising be an indication of anemia? Black and blue spots? You get black and blue spots with what? Bleeding. So if you bleed a lot, that's platelets. So bruising is platelets that are not functioning, not having enough red blood cells. So you're not going to bruise easily if you don't have enough red blood cells. Okay? But you will feel tired and fatigued. Many cancer drugs cause anemia because they attack cells that divide rapidly. Mostly your epithelial cells, because many cancers have their origin from some type of epithelia. But they also attack red blood cells because they divide rapidly. We call a particular anemia that's due to drugs that affect or radiation that affects the bone marrow, aplastic anemia. Sometimes the cause of aplastic anemia isn't known, and it might require a bone marrow transplant, transfusion of new bone marrow. Okay, so this is the bone marrow is inefficient or ineffective, not unable to have enough stem cells to replace the red blood cells. Those have been damaged or destroyed. This next slide shows iron deficiency anemia. You may have enough protein in your diet to produce the red blood cells, but if they don't have adequate iron within them, you're not going to be able to transport the oxygen. Okay? So when I was pregnant with my twins, it's kind of standard to do blood tests for anemia and stuff around, I think it was 28 weeks. And my hemoglobin, which is a measurement of the protein component of iron, came back at 8. It's supposed to be about 12 to 14. And I just thought I was tired because I had twins. So I was told to eat a lot of greens. Personally, I like kale and collard, and I just eat a whole bunch every day. That was much more fun than eating iron pills that make you constipated. So iron deficiency anemia, which could be supplied by liver, which not eating meat, the idea of eating liver was not particularly appealing, all right? Or you could have another type of anemia with a deficiency, which is called pernicious anemia. This can be due, well, it's a deficiency of vitamin B12. And sometimes people will just, I've got to go get some vitamin B12, I'm feeling tired all the time, instead of eating greens. But vitamin B12 affects the metabolism and the enzymes in producing the hemoglobin rather than the iron component. And so it can be due to a dietary deficiency. Vitamin B12 can only be obtained from animal sources. So a person who's a vegan has to take vitamin B12 pills, all right, because they're not going to be able to get it from carrots and legumes and corn and stuff like that. Now, I'm vegetarian, but I'm not a vegan, all right, so I'm lacto-ovo, so milk and eggs, yogurt, cottage cheese, ice cream. So I get plenty of vitamin B12 in my my animal products from the lacto-ovo aspect. Okay. Another cause, however, and the reason the picture of the stomach is up there, is that the stomach produces a chemical or molecule known as intrinsic factor. And this is required for the absorption of vitamin B12. If the lining of the stomach does not produce vitamin B12, Or maybe they've had a gastric bypass and have had 80% of their stomach removed. That's why the lap band procedure is better for the vitamin B12 because it just creates an adjustable band that goes around the stomach here, and it can be made smaller or larger in diameter. But the rest of the stomach, so you still have to eat small portions, but the rest of the stomach is still present. Okay, the secretions are still available. So if this is the case, this is just, pills or food. If this is the case, intramuscular injections are required. Doesn't really have anything to do specifically with the intrinsic factor, but it does have to do with vitamin B12. There was a charge of murder brought a few years ago against a medical doctor. Intent to murder was the first charge because it took the woman a while to die. But a married woman and the doctor were having an affair. They were both married. And he professed his love to her, and they'd been dating for several years. And the discussion was they would each divorce their spouses and marry. So she divorced her husband. He never divorced his wife. And she was very emotionally involved with him, and it took her quite a few years to extricate herself from that relationship when she realized that he wasn't going to leave his wife. And he kept trying to pull her back in. So she'd start dating somebody, and he'd pressure her to go back out, see him again. And she finally met somebody that she, felt she could marry, and kept trying to tell the doctor that no, I'm involved in a relationship. And one night he called her up and said, this was something apparently he did while they were dating. I don't know why she never let him do it afterwards, but anyway, I'm going to bring you over a shot of vitamin B12. 1 o'clock in the morning? I mean, I don't think I let my husband give me a shot of vitamin B12 at 1 o'clock in the morning. But anyway, so he shows up, gives her the injection, and leaves. And within 6 months to 10 months, she starts getting symptoms of HIV. And she goes and gets checked. And sure enough, that's pretty rapid. It sometimes takes 2 or 3 years. She has the early signs of AIDS. And she doesn't have any exposure. She doesn't have much of the lifestyle risk that's involved. And she is convinced that the doctor, her ex-boyfriend, whatever, was the one that gave her the virus as a payback for trying to leave him. So she called the sheriff's office, and this was a well-respected physician. She called the sheriff's office, and they're like, we're not going to go over and charge him with, attempted murder. And she kept insisting that there was no other way that she could have gotten this virus. And bless the guy's heart, he got a search warrant, went over to the doctor's office, searched his office and lab area, and found vials of blood that had HIV. And for the very first time in any legal history, they used the DNA makeup of the virus, which mutates very rapidly, and matched it to her virus. Not only that, but the guy that had come in on the same day that the doctor had gone to visit her. Not only that, he also had hepatitis C. And they matched that virus to the hepatitis that he had given her. And he was charged and convicted of attempted murder, and then when she died 6 years later with murder. And it was the first time in legal history that they had been able to use the DNA of the virus to forensically link those components to each other. As I said, it has nothing to do directly with vitamin B12, but I think about, really, I love forensic stuff, and it was, it reminds me of that every time. Okay, one more anemia, and then I'll let you guys go. Sickle cell anemia. All right, this is a recessively carried trait. It's really just a substitution of one single amino acid. Glutamate versus valine. And that is sufficient to provide the membrane instability that occurs when low oxygen is present in the blood. All right, so the blood cells aren't like this all the time, but if oxygen concentrations in the blood decrease, it causes this sickling to appear, and especially in the capillaries where the blood is flowing slowly, they break down more easily, they block the capillaries, and so there's organ deficit, function deficit, there's joint pain that occurs with this. It's a far wide-ranging type of symptoms that occur with sickle cell anemia. So this is blood typing, not typing, but blood spread that shows the sickled cells that are present. Interestingly enough, and you've probably heard this, sickle cell anemia does provide protection against the transmission of malaria, because the malaria parasite, whose life cycle occurs partly in the red blood cell, can't have its completion of its life cycle in sickle cell erythrocytes. I don't know. I think I'd rather have malaria than sickle cell, but at least there's medications for maybe not black water fever. But all right, so we'll stop there, and then on Tuesday we'll look at some of these tests and instruments that are used to test for it. One more voice recorder up here.